Hey class, this is Mr. Schmidt for Environmental Science January 21st review. First of all, you have a quiz. It's open till midnight. It's an 8-question quiz. Once you open that quiz, you'll have 30 minutes to complete it. Please have all your notes ready to go do it. Hopefully, it's just over what we've talked about all of last week, and it should be fairly straightforward. Okay, so that was the first thing to discuss. Next up, we looked ahead towards the final exam. So I looked at what you submitted for your self-assessment. So notice if people marked anything as having lots of questions, I try to spend extra time about it. If people mark that as specific questions, I ask that they text them to me and I answer those specific questions. And then of course, if you're over here, we spend less time on those topics. So as we go through the notes, I'll try to focus on those things that fall farther to the right in our preparations. So let's go back to the beginning here. It's a very long set of notes. And the first thing you'll notice is we started with a conversion. Conversions will be on the final exam. They were on a number of different quizzes. Okay, so let's take a look at how do we do this one on paper. Okay, so at 2.5, we're going to go from kilo to milli. Okay, so there are a number of other prefixes that fit within that. Yeah, okay. And so the nice thing about those prefixes is they also serve as markers for how far we need to go. So kilo to the base unit, okay, so this, by the way, was our grams. But it also could be meters or seconds or anything else. That would be what we have right here, okay? So if we just wanted to go to the base unit, then we would have moved the decimal place 3 times. But we want to go all the way over to millimeters, so we need to move it 3 more times. And so our final answer will be that 2500. Okay, 2,500,000. Okay, so that's how we do the conversion. So notice, like if you had to convert inches into miles, that would be a lot of math, multiplying and dividing, numbers I don't necessarily have memorized. Okay, but millimeters to kilometers, there's no multiplying, there's just moving these decimal places back and forth. Okay, so if we look back to our notes, we can see there's another way to represent this chart I just did. And that is through these blocks. So we can go forward or backward. We're just moving the decimal point based on where we're starting and where we're ending. Okay, so we can do that for any metric unit at any time. So you don't have to remember how many teaspoons are in a gallon or anything like that. We can just go milliliters to kiloliters. We can go to centimeters to millimeters. Okay, we can do anything we want within that. And the other nice thing about the metric system is if we have 1 milliliter of water, that is 1 cubic centimeter of water, that is 1 gram of water. So we can even go from one type of unit to another type of unit. But if you want to go from gallons of water to pounds of water to cubic inches of water, that would be hard to do. So the metric system gives us a number of advantages, and the scientific community around the world all uses the metric system, and you'll be using it in future classes and on next week's final exam. Okay, so speaking of the final exam, one whole section of the final exam is going to be writing a lab. So I'm going to pull some data from a lab you did not do about environmental science, and I will ask you some certain things about it. So let's say the lab was about pH and acid rain. Okay, well, first you would write what you already have learned in class about pH and acid rain to give the reader some background about that information. Just be 1 or 2 paragraphs, okay, complete sentences, 5-sentence paragraphs, and that would bring you down to your hypothesis. So maybe your hypothesis is acid rain is not present or detectable in the Columbia River Basin today, or acid rain close to factories is 25% higher than acid rain in the forest, okay? Or the pH of rain is 1 pH lower around factories than it is in the forest, okay? So there's a number that you're trying to get to based on what you already know about that topic. And for the exam, we're going to stick very closely to topics we've talked about this year. Okay, so once you've done that, so using this pH example, you could say, oh, we want to change how far away we are from a factory, and we want to measure the pH of the rain. Okay, so how far away you are, that is the independent variable. Okay, the pH of the rain, that is the dependent variable. Okay, and then you're going to bring that down into 1 sentence that gives the reader and the audience what they need to know about what you're about to do. So, like I said, if we move 100 miles away from a factory, then the pH of the rain will decrease by 1 point because the amount of pollution has decreased and lost its effect on the clouds. Okay, so you have an if then because sentence. All right, now you're ready to tell the reader and the audience about the procedure. So you can have numbered steps, complete sentences, numbered steps telling them what to do. So first, measure the pH of the rainwater next to the factory. Okay, well, do you need to go into more detail? Do you need to say, first, put up a little cup to capture rain next to the factory, and put up little cups every 10 miles between the factory and the forest. Number 3, collect the water samples. Number 4, test the pH of each water sample using litmus paper. Okay, so you're going to number each one and go along the ways. Okay, if things start to get confusing with words, that's the pictures and diagram part that you add into it. Okay, so the idea is that scientists in South Africa, or scientists in China, or scientists in Brazil can all go to their factories and do the same experiment and see if they get the same results. Okay. All right. So we're going to have 2 different kinds of measurements. Number 1, the ones with numbers, the pH of the water, the volume of the water. Okay. So if you have 250 milliliters of water with a pH of 6, those are quantitative measurements. Okay. In this first lab we ever did, it was the number of paper clips you put on a paper airplane. Okay. Qualitative measurements will be descriptions. The rain was a little yellow. The water in the cup was a little yellow, or insects appeared in the water. That would be qualitative descriptions. So we'll need to also note some of those things. So obviously, if animals are getting into your little cups, that's going to corrupt your experiment. So that's something you'd write about as a qualitative observation. Okay, the data is something I'm going to provide on your exam, but the idea is you're going to have a nice cluster of numbers that you're thinking about. Okay, and then we're going to have a graph. I'm going to talk about graphs in detail here in a minute. So you're going to choose the right kind of graph. Then you're going to draw it out using the correct scale and units and label and title, and accuracy and neatness. Okay, that was for that specific lab. And then you're going to analyze it. So you're going to use that data and those graphs, and then you're going to compare it and say, oh, as the distance from the factory got bigger, yes, the pH approached 7. Okay, notice when you're about 50 miles away from the factory, the pH shifts suddenly. Okay, that is a pattern to discuss. Okay, and then you have that set up and ready for the conclusion. So the conclusion will be a paragraph or answering a series of questions discussing the data has been identified and what is important about it. You're going to say, oh, my hypothesis said this, it was right or wrong, okay, and why. Okay, even if your hypothesis was wrong, it can still be a very powerful conclusion and an important piece of research. Okay, then you're going to discuss the results also, and also support the research with your introduction. So you read about how factories cause acid rain, and that's what you put in your introduction, and then you're like, oh, my data, says it does so, but to a different degree than was thought by those people. Okay, and then you're finished off with the big piece here, the one that gets scientists to have a long-term career. They're going to say what they want to do in their next experiment. Do they need to measure farther than 100 miles? Do they need to do more steps in between the 10-mile mark? Do they need to go to other countries and see if they can reproduce this? Okay, what would be their next step is what we're trying to ask here, and that's where you need to finish your conclusion. And like anything in this entire lab, everything should be neat, should be complete sentences, should have correct grammar, and clear communication. So one of those things I mentioned was pie graphs. So pie graphs will be in the lab, or some sort of graph will be in the lab. And I'll also ask you multiple choice questions about graphs. So we have one whole part of the test was just the lab, and writing up a lab during a lab practical. The other part of the final exam is going to be a multiple choice test. And a good example of those questions is going to be your practice. All right, this is all the questions off of the old quizzes. So if you wanted to look at this, it's not going to be due, you don't have to turn it in, but it is going to be the best place to look for examples of questions and the style of questions I'll ask. So if I asked water changing from a liquid to a gas, maybe on the final exam I'll say, what's when you change from a gas to a liquid? What about a solid to a gas? Okay, so you see the style is right there. I might just change a couple words to see if you know all the surrounding information. So one of those questions, or a series of questions I'll be talking about or asking about, is the type of graph. So we have three big categories that we did in this class so far. You'd had your pie graphs, we're looking at percentages. Percent of carnivores versus herbivores. Percent of people who like favorite soda. Percent of flowers versus grasses versus pine trees. Okay, those are good percent type questions, make good pie graphs. Okay, bar graphs work well for totals. So total number of birds on the island. So you'd have like eagles, cardinals, seagulls. Each one would have their own category and their own totals. So again, this is one where you'd want to think about when you're doing it inside the lab, which one's doing a better job of investigating your hypothesis. On the multiple choice part, I'll tell you which one I want. I want a percentage or I want a total, and then you're going to show me which graph you're going to do. And then the third type that we focused on this year is the line graph, which is the change over time. So if you plotted the height of plants every day, that is a good line graph. Also true is acceleration or velocity or time that physics does. Okay, those students are going to see a lot of line graphs as they do distance versus time, velocity versus time, acceleration versus time. There's a lot of change over time that happens in those categories. So next up, we're talking about open, closed, or isolated systems. So let's look at both of these. These both hold hot water. Okay, they are both have the lid on it and they're sealed closed. If we put this green one on the stove and heat flows in and some of the heat radiates out, that becomes a closed system. No water came in or out, but the heat came in or out. Now, if steam starts shooting out the lid, then now it's an open system. Compare that to this one, which is basically the original hydro flask. This is the thermos. You have an insulated layer and a closed lid. So no water can go in or out, and basically no energy is going in or out. That makes it an isolated system. Okay, so to put that into words, what we just saw, open systems, matter and energy move in and out. So we see that a lot in biology and a lot in environmental science. Cells take nutrients in and send off heat, and sometimes they send off light or other type forms of energy as well. Okay, closed systems, maybe we're looking at the inside of a stomach or a teapot or something where not stuff for that specific point in time, no matter is going in or out, but energy and heat and light can go in or out. Okay, that would be a closed system. And finally, our isolated systems, no matter or energy move in or out. So if we think about a cooler or a hydro flask or any of those types of things, it's sealed and insulated, no matter, no energy, it's all isolated. Okay, so now we can think about, once we're inside the systems, we can think about phases of matter, solids, liquids, and gases. So obviously this affects water. This is part of the water cycle. We'll talk about the other cycles later, but the water cycle is such a big part of the entire environment. And the ones we're most familiar with is evaporation and condensation. So things going liquids to gas, gas to liquid. Okay, those are the most common, but we can also have other ones that happen in nature. If you go up to the top of Mount Hood, it's a bright sunny day. The snow goes straight from solid snow into gas state. It never melts, it never evaporates. It just goes straight from solid to gas based on the temperature and pressure that it's around. The other one that's very common is transpiration, which is the release of water from plants. So it's not completely in its liquid state because it's inside a cell and it's mixing with all these other things. Okay, so it's not completely a pure water in its liquid state. It's a mix of other stuff, but the water itself can leave, and it's leaving through the holes, the spores in the plant. Okay, and that sends a great deal of moisture into the atmosphere. When we think about all the trees and all the plants on the entire planet, it's a lot of moisture coming through the roots, through the whole plant, and out through the leaves. Okay, and that's transpiration. And those are all parts of the water cycle. Like I said, we'll come back to the cycles here in a little bit. Next up, we have the biomes, which are the type of environments that is defined by the organism that live there. So there's two different ways I can ask questions here. I can say something like, which biome is known for having almost no precipitation. Deserts have almost no precipitation. Okay, that's one way I can ask it. The other way I could say it is like temperate forests. What kind of plants are common in temperate forests? And you would have to say, well, large trees like oaks and maples and deciduous trees are in temperate forests. Remember, this is all going to be multiple choice, so hopefully it's clear categories here. Marine is a saltwater environment. Okay, freshwater environment, well, that's what we saw in our bottled biosphere. Okay, and so there's going to be very little salt, but there's going to be a lot of algae, maybe some moss, fish, those types of things. Okay, so we be prepared to answer that on either way, and look at the exam and the old homework if you need more examples. So what makes one biome different from another? Well, number one, the vegetation. That's kind of the indicator that we end up looking at. But that vegetation is normally caused by the latitude, or how far north or south it is, and the altitude, how far up it is. So if we think about the top of Mount Hood, that is a tundra environment because it's at very high altitude and very cold and has very specific vegetation because of it. Okay, that's going to become similar to the South Pole. That is very cold, has very little vegetation because of it, because of where it is north to south. So next up we see biodiversity, which is the variety of life and species found in an island or any habitat. Okay, so that again, latitude is going to be a part of it. Habitat size is going to be part of it, so how big of the island it was, and the amount of time that that habitat has been present. So if the lava just formed and that island just arrived, it's going to be very little biodiversity. But if that island has been sitting off the coast of Florida for thousands of years, there's going to be a lot of opportunity for different things to move there, to thrive, to evolve, and so it's going to have a lot more diversity. And that's going to be based on the optimal range, which is the abiotic factors that produce the highest possible population. So if we think about the things with the highest optimal range, that'd be like the Amazon rainforest. Okay, what are the abiotic factors, the non-living factors? Well, it gets rain. It's close to the equator. It's a very large piece of land. There's lots of nutrients moving through the Amazon River. So that has all sorts of abiotic factors, non-living factors, and that's going to turn around and feed the opportunity to have a diverse environment. So when calculating biodiversity, we can look at species richness, which is the number of different species in a community, or the relative abundance, which is the number of individuals in a species divided by the total number of individuals in a species. Okay, so we're going to do the number in the present versus the total number. And that also gives us the keystone species. Okay, where the keystone species is the species critical to the functioning of an ecosystem. So we talked about like otters are important for the ecosystem off the coast of California and Oregon, the kelp forests. The jaguars are a keystone species to the jungles. There are certain species that if that one species disappears, then everything else changes radically and there's no stability. But if this one species is present, then what it eats and what they eat and what the environment is stays very constant. Okay, so that is the keystone species. So we already talked about water cycle. Now we're talking about the other cycles. And this, by the way, was one highlighted by students asking for more details. Carbon cycle is the majority of carbon is found in the oceans and soil. Okay, And photosynthesis is the key driver of the carbon cycle. So that's carbon dioxide and water coming into plants, coming into the plant cell, going in through the individual parts of the plant, which we don't have to worry about. Okay, and then it gives off glucose and oxygen, glucose being the most fundamental sugar, which we end up eating. Also important in the carbon cycle is fossil fuels such as coal, natural gas, and oil. Okay, so those are all coming out of the soil, by the way. And then the other key cycle is the nitrogen cycle, which is nitrogen's found in air. Not very exciting in air, it just kind of goes in and out of our lungs without doing anything. But it's important for plants. Plants need it somehow into their roots so that they can make all these important molecules, okay, like DNA or protein. Those all need nitrogen in a different form. So in order to turn nitrogen gas into nitrates that plants can use, specific organisms and instances can make that change. So the way it gets changed from nitrogen gas into nitrates is specific bacteria on the roots of legumes, which is why beans are so important. Beans and their roots really help the soil. Okay, blue-green algae in the ocean is also very important for the same reason. And interesting, lightning can also turn small amounts of nitrogen gas into nitrates. Okay, and then I think we're going to end with this one right here. Tropic levels, that's looking at energy levels. So we see the sun comes in as our primary energy source on our, what is essentially a closed system. Earth is a closed system. Energy is coming in, but no matter is coming in or out really. So energy from the sun comes in, hits Earth. Plants take that. Plants are a category called autotrophs, or the producers, which is the bottom layer of the tropic levels, the energy levels. And then you get things like rabbits eating the plants. So they are primary consumers. They are the next level up on the trophic level. They're the herbivores. And then you get things like snakes eating the rabbits on that third level, considered secondary consumers. Those are one type of carnivore. And then you'll get things like eagles eating the snakes as top level consumers and tertiary consumers. Okay, and then that also gives us a model for not just the energy going through, but also the nutrients. So the nutrients at the base level in the grass will keep getting more and more concentrated until you get to the tertiary consumers, the top level carnivores. And toxins can follow that same path, so that any little bit of toxins in the grass may end up overwhelming the eagles, because all the toxins from all those grasses may end up in that top level carnivore. Okay, but when we're talking nutrients, those nutrients then get trapped in all these different individuals, and they are broken down by the recyclers, the scavengers, the detritivores, and the decomposers. Okay, so the detritivores are things like dung beetles, and I would put worms in this category, breaking down dead tissue and waste. Scavengers, by the way, are things like raccoons. And then decomposers are like fungi and bacteria. So we see things like mushrooms, breaking them down and recycling the system. Okay, and then that's where we're going to stop for today, and I'll talk more about these next week.